Welcome, online listeners and viewers, and thank you for joining this Science AAAS webinar, Building Things in Brains, Chemical Constructions for Analysis and Discovery. My name is Jackie Oberst, and I'm the Assistant Editor for Custom Publishing here at Science. I'll be moderating today's webinar. Joining us today as guest speakers are Dr. Karl Deisseroth from Stanford University in Stanford, California, and Dr. Mark Brown from Andor Technology in Belfast, Northern Ireland, and whom we will learn more about shortly. Before we get started, I'd like to walk you through what's being currently seen on the screen for our online viewers. At the top right, you'll find a photo of today's speakers and a View Bio link, which you can click on to read more details about their background and research. To the right, you'll also see the Resources tab, where you can find additional information about technologies related to today's discussion and a PDF of the slides. After the speaker presentations, we will have a short Q&A session, during which we will answer some of the questions submitted by our live online viewers. So, start thinking about some questions now and submit them at any time by clicking the Ask a Question tab, also on the right, typing the question into the message box, and then clicking Submit. If your question wasn't answered, we will do our best to reach out to you after the program. You can also log into your Facebook, Twitter, or LinkedIn accounts during the webinar to post updates or send tweets about the event. Just click the relevant icon at the bottom left of the screen. For tweets, you can add the hashtag #ScienceWebinar. Finally, thank you to Andor Technology for sponsoring today's webinar. And now, let's begin. Unmatched in its complexity, the brain may be the final frontier of biomedical research. In this webinar, Dr. Karl Deisseroth, a neuroscientist, bioengineer, and psychiatrist, will discuss the principles and practice of a powerful new way of interacting with complex biological systems. Over the past 7 years, an in situ chemical synthesis approach to biological systems has emerged from his laboratory and has been further developed and applied around the world, in which functional materials are assembled within tissues such as the brain, either constructed throughout the intact tissue, hydrogel-tissue chemistry, or HTC, or genetically targeted to cell types, genetically targeted chemical assembly, GTCA. The resulting hybrid materials are endowed with diverse capabilities, including anchoring and labeling of RNA and protein, in situ sequencing, as with STARmap, tissue transparency, reversible size changes, swelling, expanding, or shrinking, contracting, and electrical insulation or conduction. In this talk, we will discuss the principles and practice of this powerful new way of interacting with complex biological systems, which involves reimagining and transforming the system with integrated optical, genetic, and chemical methodologies. Without further ado, I'm very pleased to introduce our first speaker to you now, Dr. Karl Deisseroth from Stanford University in Stanford, California. Dr. Deisseroth is the D.H. Chen Professor of Bioengineering and Psychiatry and Behavioral Sciences at Stanford University and an investigator for the Howard Hughes Medical Institute. He continues as a practicing psychiatrist at Stanford with a specialization in affective disorders and autism spectrum disease, employing medications along with neural stimulation. Over the last 16 years, his laboratory has created and developed optogenetics, hydrogel tissue chemistry, beginning with Clarity, and a broad range of enabling methods. He has also employed his technologies to discover the neural cell types and connections that cause adaptive and maladaptive behaviors, and has disseminated these technologies to thousands of laboratories around the world. Welcome, Dr. Deisseroth, and thank you for joining us today.
Great. Thank you, Jackie. It's a real pleasure to be here to share our ideas, thoughts, advances, and to engage with the community. This is an exciting time in not just neuroscience, but in general, the study of intact systems. And what we're discovering is that chemical approaches, chemistry, is turning out to be very powerful in helping us to exchange information with complex biological systems. I'm a neuroscientist, so what you'll see today is focused largely on the brain, but these principles, as you'll see, are quite general. Now, what I'll do is first talk about how we build function into brains. I'll start with the first half of the webinar, talking about how we use chemistry and chemistry-related tools to endow function in two parts of the brain, particular structures, particular components. And then in the second half, I'll talk about actually building new kinds of structure in the brain and how that is useful. We've been working on endowing cells with specialized kinds of function, and this is called optogenetics. We have developed an array of methods that, when brought together, allow you to turn individual types or even individual cells on or off in living, behaving mammals. And we do that by delivering single proteins. These come from the microbial opsin gene family. These are light-activated regulators of ion flow. Because they're single genes that we can deliver using genetic or anatomical tricks, we can then deliver light broadly or focally and cause specific changes in activity. And this has turned out to be quite useful, but what's really exciting from the chemistry standpoint is what has made it especially powerful, even fundamentally so, has been diving into the structure, the chemical structure of these proteins that are encoded by the microbial opsin genes. What you see here are three crystal structures from our laboratory and our team, and this has allowed us to understand how the channel rhodopsins work. These are light-activated regulators of ion flow that form pores, holes in the membrane that open in response to light. This was the initial one with our team. We got this structure in 2012, and it's a cation conducting channel rhodopsin. These two structures are anion conducting channel rhodopsins, and these structures we described in 2018. And having this level of atomic level resolution has enabled us to create new kinds of function, fundamentally new kinds of function. By engineering and retooling and redesigning the protein based on placements of atoms, for example, along the ion conduction pathway, we've been able to create a whole host of new kinds of logic. You can see in the middle of this, the middle column of this slide, 4 fundamentally different kinds of light spike logic, or photon spike logic. Very fast spiking at hundreds of hertz at the top. These individual sawtooth-like deflections are action potentials, the currency of information flow in the brain. Fast blue light spiking. The second one down shows red light-driven spiking. The third one down shows a bistable mode of operation where cells are flipped into and out of excitable states. And the last one shows bistable inhibition, with cell being flipped into and out of a refractory state. All this done with single designed microbial opsins our ability to do this arose from our structural understanding. And this work was carried out with a very important team of colleagues and collaborators from around the world, and some of their names are shown here. Now, what this has enabled us to do more broadly is do something that Francis Crick called for back in 1999. He said a fundamental challenge in the brain is to turn types of neurons on or off in a rapid manner, and he even thought the ideal signal would be light in 1999. And he didn't know how to do it. He said it would be far-fetched. Of course, now we can do it, but Francis Crick, in his very inimitable way, had put his finger right on the core of the problem. And being someone coming from the biochemistry and genetics system, this is a key principle that biochemists and geneticists are very familiar with. In the study of intact systems in biology, like the brain, we also seek to test hypotheses, just as a chemist or a developmental biologist or a biochemist might. And brains, other complex systems, are complicated. They're heavily interconnected. There's feedback, there's redundancy, there's nonlinearity. But all of this is not different from genomes or developing organisms or proteins in any fundamental way. So we seek that same level of control. And just as a researcher studying a protein or a gene will inhibit or knock out that gene or activate and deliver it, we want to do the same thing in complex systems like the brain, where the equivalent of the activity of a gene or protein is the activity of precisely defined neural circuit elements during behavior. And that allows us to do hypothesis testing just as any other field of science can do. In some ways, those of you who know developmental biology will be familiar with the power of this approach. This is the Antennapedia mutation, where legs grow where antenna should grow, and this is seen on the right, where the legs are growing from the fly's brain. Identifying the single gene that allows this program to happen was truly transformative, eye-opening, and it required being able to adjust the activity of a specific gene. And of course, even today, we don't fully understand all the downstream activations and inhibitions that together give rise to exactly how a leg is formed, but we understand the key principle, which is a single gene, if targeted in the right way, can give rise to complex function. And we see this with optogenetics too. I'll just give a couple quick examples. This is delivering a channel rhodopsin in a virus to a brain structure called the bed nucleus of the stria terminalis, or BNST. This was work from Sung-Young Kim and Abhishek Adhikari and others in the lab. And what you can do after injecting, as the syringe shows, a channel rhodopsin gene into one brain region, you can deliver light by fiber optic using our fiber optic interfaces that we developed and described in 2007. You can deliver those, as the little squiggly blue arrows show, to downstream structures across the brain. And you can thereby recruit cells defined by origin and target, because the only light-sensitive element in the downstream target region, the VTA, the lateral hypothalamus, or the parabrachial nucleus as shown on the bottom, are those projections picked out by origin and target. And we were able to deconstruct the state of anxiety into its component parts, with the valence of the state being controlled by the VTA, the risk avoidance, the behavioral changes being controlled by the lateral hypothalamus, and the respiratory or physiological changes being controlled by the parabrachial nucleus. And this has turned out to be very general. We've seen also a number of labs around the world apply this method. Catherine Dulac's group at Harvard had a beautiful paper in 2018 deconstructing parenting, if you can believe that, using these same exact principles, same tools, and finding that projections from the MTOA to different downstream structures across the brain governed different aspects of parenting, going out and getting the offspring versus grooming for them and actually caring for them. And they use the same principle and the same turning on or turning off using optogenetic tools. So this principle of endowing structures with function is quite fundamental, and being able to do that in a targeted way has been very powerful for neuroscience. I'll give you one other very recent example through which endowing structures with function has given us important insights. And this was work from my lab that just emerged a month or so ago. Now, what we have here is a very deep question, and a question that seems almost inaccessible to neuroscience, which is how to understand something like dissociation. Some of you might have heard of dissociation. This is a state that happens when one's sense of self is separated from one's body. And I'm a psychiatrist. A lot of you may have experienced something like this. It's very common after trauma, very common in psychiatric diseases, but many of you might never have experienced this, and it might seem like a very intractable concept for scientists to address. But optogenetics has given us a route into this, and I'll tell you briefly about how this worked. So we imaged, using wide field optics, a living mouse brain, and using wide field optics and genetically targeted calcium sensors, we were able to observe the activity all across the brain, the surface of the brain, in a very unbiased way. And we could do things like give agents, pharmaceutical agents, that are known to cause dissociation. And we saw something very striking with this global unbiased approach. We saw with ketamine, we saw an interesting oscillation appearing in one little patch of cortex called the retrosplenial cortex. And we were pretty interested in following up on that. We didn't know why that was happening. It was very specific just to retrosplenial cortex and not other regions, as you can see on this slide. And what's more, it was, as you see in the lower left of this slide, it was also seen with other dissociative agents like PCP and MK801, but not with non-dissociative agents like memantine. And we delved into this further. You might say, well, that's an interesting phenomenon, but what does it mean behaviorally? And here, it took us several years to sort this out. How do you assess a question like this in a mouse? But we can give a warm stimulus that an animal has two types of responses to. First, there's a reflexive withdrawal of the paw in response to the stimulus that's too warm. That's very quick and automatic. And then there's a longer what we call an affective or emotional response, where the paw is licked to cool it, and there's other kinds of more complex movements to escape. And what we found, as you can see on the right, is that at doses of ketamine, 25 to 50 milligrams per kilogram, that caused the oscillation, as you can see on the lower left in C, if you look at the upper right in B, those doses of ketamine did not cause a change in the reflexive flicks, the immediate stimulus detection response, but abolished the more affective or emotional response. And for us, this was a pretty interesting correspondence to what people see in dissociation, where stimuli are still known to occur, but people experience a sense of being separated from their body, and so they don't care about it. And this is why ketamine in some conditions can be used in people who, as a therapeutic agent, in people who are experiencing certain kinds of pain. So this was a very interesting correspondence. We dived into this in more detail. We saw that PCP had exactly the same pattern. It preserved the reflexive response, as shown in yellow on the leftmost top panel, but abolished the emotional or affective response, the second group over. As you can see, the yellow bar is almost completely at the bottom. And then we were able to use optogenetics to come in and say, what if we provide this exact type of oscillation that we see here, and we can intersect that function with structure, because we could use genetic tricks to target a microbial opsin gene to just these particular deep retrosplenial cortex layer 5 cells, turn them on or off in a particular rhythm. And what we saw was a behavioral effect that corresponded to what we'd seen in response to ketamine. If you look at the middle of the bottom row, you can see with our rhythmic stimulation, the magenta bar, that the reflexive foot withdrawal flicks are maintained, but there's a significant reduction in the next bar grouping over in response that we see the licking or the affective behavior is significantly reduced in response to the rhythmic stimulation. And this was not seen if we targeted this intervention to somatosensory cortex, SS, compared to retrosplenial cortex. So this was very interesting. We delved further into the mechanisms underlying this, and we tested a number of possibilities as the paper details. I'll just focus on one here. There are, as we noted, very interesting patterns of expression of particular ion channels in this patch of cortex, and just focusing on the HCN1 channels. These are known pacemakers. They generate rhythms or oscillations in the heart as well as in the brain. And they are very highly expressed in these deep retrosplenial neurons, much more so than surrounding cells. And using genetic tricks, we knocked out the HCN1 channel just in those cells, and what we found was that prevented a number of very interesting things, effects of ketamine. If you look all the way at the bottom middle of the slide, with the HCN knockout, we saw a loss of the ketamine-induced oscillations in retrosplenial cortex that you would normally have seen. And we also saw a restoration of the affective or emotional response, as you can see in panel F, the two groupings of bars at the left. The second one over shows a restoration of the affective response in these HCN1 channels. And this was very interesting, that you could restore the affective response in the presence of ketamine just by taking away this channel in these particular cells in this one patch of cortex. So very interesting, both providing and taking away rhythm-generating mechanisms was interesting. But of course, we wanted a more integrative understanding of what the whole brain was doing. And here, optics faces certain challenges. Because of light scattering, you can't see deep all the way into the brain. You can just see the surface of the brain optically using conventional tools anyway, or at least advanced wide field optics as we used. But using brain-wide electrophysiology, we could get deep. And this was done with these long shank, high contact density recording electrodes called neuropixels probes. Will Allen in the lab had described this for us in a collaboration with Li Chun Lo's lab in 2019, having different trajectories of the neuropixels probe through the brain of a mouse, one probe per animal, but having a very temporally precise task so we could align all the different results from different animals together. And what Will did was a very simple experiment on the face of it, which was, let's look at a very simple motivated behavior, drinking water when thirsty, and let's record from all the neurons across the brain. And let's see, first of all, what is the brain-wide representation of this very simple, important, primal motivated action. And then let's see what happens when we turn up or down the thirst optogenetically. Are we recapitulating that pattern, or are we doing something else entirely? So that was a very interesting and important question. And so what you see here on this slide is the task that Will set up for the mice. These three vertical dotted lines are 3 times in the task. There's an odor that appears at time 0, and the mouse is trained to know that one odor means water is going to come later, and the other odor, the no-go odor, means water won't be coming later. And the mice learn very well to lick in response to the go odor. You can see this behavior in the lower left. All these little black ticks are the animal licking as you go from the first trial 0 to late trials in the behavioral epoch. You can see the animals licking very reliably for the go odor, but not for the no-go odor, until at the very end, the animal stops licking entirely. It's sated, it's no longer thirsty, so it no longer responds to either the go or the no-go odor. So what's going on all across the brain? This is the behavior. Well, what we see is something quite amazing. There's a brain-wide engagement in this task, and the first of these colorful panels on the left shows all across the brain these color-coded brain regions in this rainbow bar. You don't need to worry too much about the specific structures, except they're all across the brain. And these 3 dotted lines correspond to the 3 moments in the task as shown at left. And you can see all across the brain, within each brain region, within each color-coded region, we've rank ordered the individual neurons by order of the time at which their activity peaks. And red means more active, blue means less active. And so each brain region has its complex representation of the task as it proceeds. And really, this is a very powerful brain-wide representation. More than half of all the recorded cells were significantly task modulated in thirst, and that was true for the go odor, but not the no-go odor, and it was true when they were thirsty, but not sated. So then, a very interesting question, when we endow particular structural elements with function, optogenetically, what happens to this very complex brain-wide representation? Is there any chance that we're actually able to recruit this beautiful and complex brain-wide pattern optogenetically? And so here's what we did. Using optogenetics, we conferred activity onto the input 2 thirst neurons, and these are in the extended hypothalamic complex. These are subfornical organ neurons that are projecting to the MNPO nucleus. And we had identified previously these as being inputs to thirst neurons. And in the lower left, you can see the animal behavior. The animal's licking for water during the task, then becomes sated. But then at the lower left, the blue light, we deliver this optogenetic stimulus, and the animal behaviorally begins to act as if it's thirsty again. So really interesting. This is something that shows these neurons are causally involved in thirst. By the way, we know when we inhibit these neurons, thirst and water-seeking behavior goes away. What is the brain-wide representation in this case? Well, amazingly, it's very similar, resembles closely the natural thirsty pattern. So at the left is a thirsty mouse, its brain-wide pattern. The second column over is the sated pattern. Third column over is the sated mouse, but getting the optogenetic stimulus, and you can see we recruit this brain-wide, very complex, naturalistic pattern, and then that goes away upon washout. Okay, so this shows what we can do in terms of collecting a brain-wide representation. Can we bring this back to that dissociation experiment and get a sense of what the brain-wide pattern is? And here, this very much evokes these complex developmental biology experiments where a single, well-targeted intervention recapitulates a very complex and beautiful program. So what Ethan Richmond in the lab did was he brought these neuropixels probes, and he had designed a 4 neuropixels probe, so even more power than the prior approach. 4 simultaneous probes in the animal. And what he saw was something very interesting. The ketamine state, as shown on right, a very striking pattern popped out. So these are correlograms. You can see on the left is pre-ketamine, the right is post-ketamine. And there's if you look at the RSP, there's a very pronounced red square where RSP meets RSP. That means it's increasing its oscillation. It's more correlated with itself. But then this cluster, this 3x3 square at the lower right, are thalamic nuclei. And you can see some of them are correlated with each other and retrosplenial, as shown in red. But some, particularly the AM nucleus, is dissociated. It's coming into, as we can see directly in the raw data, into an inverse correlation, as shown on the top. The anteromedial thalamus is now forced into a rhythm that's out of phase with retrosplenial, whereas the other nuclei, the anteroventral and lateral dorsal, are in sync with the retrosplenial. And these latter two are known to be heavily connected by wiring to the retrosplenial cortex, whereas AM is not, more involved in frontal projections rather than posteriorly to the retrosplenial. So we can actually see right before our eyes a functional dissociation happening. One part of the brain is being brought into a rhythm, another part is being forced out of that, and this may help us interpret the nature of the dissociated state. Now, this is very interesting in mice, but you may ask, is this relevant to human dissociation? And in the same paper, we described brain-wide recording, analogous in some ways to the NeuroPixels probe experiment, in a human being. And this is a human being who came to the Stanford clinic with epilepsy, and in particular with an aura prior to seizure that was dissociative. This was a patient who had dissociative experiences just before the seizure. And in terms of in the process of the brain-wide diagnostic recording, this is the normal clinical workflow that happens. What our team found was that there was a rhythm that was appearing specifically during dissociation, very well described dissociative experiences that this human being was able to verbally describe. And when this happened, there were, in these structures that are homologous to the mouse retrosplenial cortex, pronounced 1 to 3 hertz oscillations around 3 hertz that were very much reminiscent of what was happening in the mouse. And as a final stage in clinical mapping, stimulation is done in the human beings, and what was found stimulating in these oscillating regions evoked, as you can see on the right, classical dissociative experiences. So what this has shown us is that we can bring very powerful, single protein, well understood at the chemical level tools, and get to a very high level understanding of things as complex as anxiety, as parenting, and dissociation by endowing well-targeted elements with function. I'll touch on one other example before we get to some of our very latest chemical tools. And this is an important one because it gets to the nature of sensation itself. What this team of talented and creative students and postdocs in the lab did, Jim Marshall, Yoon Kim, Tim Machado, and Sean Kieran and colleagues, was discover a broad range of methods that allowed us to play in patterns of activity at the single cell level. Everything I've talked about so far has been cell type level. Now we get to the single cell level and turn on or off patterns, ensembles of individually specified cells. In the upper left, you can see this required our identification of a new channel rhodopsin called carmine. The arrow shows this. It's a quite unusual but very potent and very light-sensitive channel rhodopsin that let us play in complex patterns of activity at the single cell level. And we had mice that were trained to discriminate, as you can see in the lower right, vertical or horizontal bars of light and get a reward. And we also were imaging the brain while they were doing this, and we could see which cells, as you can see in the upper right, naturally responded to vertical or horizontal visual stimuli, and then we could optogenetically come in and stimulate those exact single cells. And what we see at the lower right is that the animal could respond perfectly well to the correct orientation of light and get reward visually, but also optogenetically. If there was no visual stimulus at all, but we optogenetically stimulated the vertical cells, the animal would respond appropriately. And we were able to play very interesting games that were very interesting insights, seeing how few cells you could stimulate in order to get this response, for example. Just to give you a flavor of what this actually looks like, here are some of those cells being stimulated. The red circles are denoting the cells that we've targeted using the spatial light modulators that allow us to play these spots of light onto the brain. These are very large, special spatial light modulators that we designed and commissioned and put into place to use this approach, and some of them are shown at the bottom. Now, bring up this experiment. I won't delve more into this anymore here, but just to highlight a motivation for some work we're doing going forward. As you look at this pattern of flashing cells, you might think, well, this is great. We can understand where these cells are. We can turn them on or off. We can see what are the quantitative parameters that give rise to perceptions, cognitions, and behaviors. But you might say, I really want to know a lot more about these cells. And this, as you can imagine, different behaviors, different actions, things that are clinically relevant, things that are of fundamental basic science importance. You want to know a lot about these cells, right? You want to know that they're there and what pattern they're in and what their positioning in a microcircuit is, but you also might want to know their brain-wide wiring. You might want to know their detailed molecular identification as well. What's their transcriptome, for example, of individual cells that you're seeing in that same animal having a natural and causal role. And here's where chemistry has been really useful for us. We've developed methods of turning biological tissues into hydrogel tissue hybrids that allow us to preserve structure and function, and even at the single cell level, transcripts, and we can see wiring, we can see a complement of expressed genes, and all of this while retaining 3D structural information. And this approach is building structures in brains, not just function, but actual physical structures. And this hydrogel tissue chemistry approach comes in many forms now. The initial form we described is called Clarity. We and many others have devised many variants since then. Basic principle is shown here, starting at the upper left, A, going over to the upper right, B, and then clockwise to C and D, shows you the different steps in the process. You take an intact piece of tissue, even an intact brain, for example, and you build, as it shows in the upper right, within that structure in a specific way, a triggered way, everywhere within the tissue at once and within the cells at once, a scaffold, a hydrogel scaffold is shown by the wavy green lines, and you specifically anchor molecules of interest, for example, transcripts. RNA is shown by the wavy red or magenta lines, or proteins. And then you can, once you've got molecules of interest anchored to your new scaffold, you can transform your tissue. You can remove lipids. You can digest proteins. You can do whatever you like to it, change its size and shape. You can start sequencing the transcripts. And as in the lower left, what you can see, what you're left with is really a transformed piece of biology that's become very accessible and tractable. The first steps toward this process were very interesting. I described this in a Scientific American piece. What we did is we started building, assembling filaments and structures inside tissue. We asked what genes can we deliver that could help us build structures inside the brain. And so we started building using a keratin, a very robust fibrillar protein from skin, and we found we could create keratin structures and filaments inside the brain. And that's just one of the very earliest examples, starting just about 10 years ago. The hydrogel tissue chemistry approach is summarized here, and I'm going to zoom in on this slide in a moment, but overall the idea is to create this scaffold, to anchor molecules of interest to it, and then create a tractable structure going forward. What we do, and you can carry out that anchoring in a variety of different ways now. We can anchor both polypeptides and nucleic acids to the hydrogel scaffold. A lot of this is described in the review, the exact chemistry. Please have a look if you're interested. Once your construction is complete and you can create this gel from many different kinds of monomer, some of them are shown here in the green box that have been shown to be useful. You then have a transformed tissue. You can trigger polymerization from the monomers and create your assembled gel. This creates this sort of backbone, as you can see in the upper left, the basic Clarity backbone. Many variants have come out since then, very similar fundamental backbones, and this allows you to do a number of very powerful things. This structure is robust. You can wash on or off probes over many cycles. You can do many rounds of labeling, dozens of rounds. Because this is all aqueous and non-organic, native fluorescence is preserved, and so this is relevant if you want to preserve the fluorescence of native fluorescent proteins as part of your labeling. There are, as you can see, reversible size changes that happen associated with this process. And you can even sequence within this intact tissue, and I'll show you a couple examples of this. The STARmap method, as shown in blue, is what I'll get to in a moment. But even before we get to that sequencing method, Emily Silvestrock and her colleagues in the lab found that you could use hairpin chain reaction, or HCR, a way of identifying individual transcripts in small numbers, you know, a handful on order 5 to 10 at once in tissue, and we devised ways to make that particularly compatible with the hydrogel tissue chemistry method called Clarity. But you may want to get far beyond 5 to 10 carefully selected genes. You may want to take a broader, more integrative view. And this was, you know, we had started moving toward this goal in zebrafish. A number of folks from our lab, Matt Levitt-Barron and Aaron Andelman and others, had been developing and applying ways of using this. In particular, the one from Matt Levitt-Barron, Multimap, allowed us to create these very complex single-cell level interrogations where we imaged activity of cells during behavior in the fish, and then we did very rich molecular phenotyping of individual cells and aligned or registered those two data sets. So we could say, okay, as you can see right in the middle of this slide, all these different colored labels, oxytocin, arginine vasopressin, or peptide Y, VIP, and so on. We could do molecular interrogation of the same cells that we'd observed their activity patterns during life. But again, this was a subset, carefully selected set of molecular targets. If you want to look at, maybe you want to look at dozens or hundreds or even more than 1,000 genes per cell, what you actually can do that using the STARmap method. It's another form of hydrogel tissue chemistry developed by Xiao Wang, and described this with Will Allen a couple years ago. And I won't go into all the details of the method, but you can see in the upper right here, it's a HTC method with the principle here anchoring a rolling circle amplified nanoball of DNA in place, and that allows you to carry out low background, high resolution optical sequencing of barcodes that have been used to label individual transcripts. And so you can take this constellation of cells that you might have observed, like I showed you in that flashing visual cortex map, where you see a lot of interesting cells, but you want to know a lot more about them, as shown in lower left, lower right. You can use this method to transform them into a really a transcriptomic level, single cell identified collection of elements. And here's an example of those little nanoballs in a single cell. This is a single cell, and this is the level of resolution and labeling we can get. Ethan Richmond in the lab has been really accelerating this, and he's made clear that you can do registration, even in mammals, of the in vivo single cell resolution 2-photon to ex vivo STARmap preparations. On the right, you can see this yellow. Where you see yellow, that's a conjunction at the single cell level of red and green, pre and post life. And this is the fundamental goal. Again, this is far more than the handful of carefully selected genes that you could do with prior methods. Now with STARmap, you have this flexibility of accessing, enormous numbers of transcripts per cell. And those don't have to be just bits of molecular identification. You can also get connectivity, brain-wide wiring information. using that same method. You can inject barcodes across the brain that are taken up because we use viruses like this AAV retrovirus, and there's a few forms of that one can use, but you can deliver barcodes by virtue of connectivity, by wiring, and cells that project different regions will take up those barcodes and bring them back to the cell, and those can be part of what you sequence in your STARmap method. So this gives you, rich molecular information, also wiring information, and registerable to the natural pattern of activity during life, and it all arises from this core hydrogel tissue chemistry method. Now, the last thing I want to talk about is not just building function and not just building structure, but can we build functional structures that are even genetically targeted? And as hydrogel tissue chemistry it's a global approach. It gives you single cell resolution information, but you're not constructing something in a cell type specific way. You're constructing it within all the cells at the same time. So is there a way to do what you might call genetically targeted chemical assembly of functional materials? And this is a very fundamental concept that you might imagine could be possible. And in a very exciting collaboration that's been going on for just about 7 years, Jinan Bao and myself at Stanford have been developing this method and showing that this kind of thing can be done. And what you could imagine different ways of making this happen, as indeed we did. We brainstormed about this quite a bit. You could imagine targeting components of a reaction. You could imagine targeting enzymes or catalysts of the reaction. You can imagine targeting reaction conditions even, pH or redox or something else. And what you're not targeting, you could deliver globally, as long as you create a necessity, a conjunction of all the required elements in a genetically targeted way. And so what we were able to do was, in fact, make this happen and create the synthesis of functional polymers on individual cells. We have a targeted way of doing this. It's described in detail in the paper, but in this case, we used Apex-2 to really affect the reaction conditions. And this allowed us to create a polymerization of polyaniline from aniline monomers and dimers, as shown here. And you can see these images in the upper right, in the phase contrast view, the dark labeling is indeed the assembly of a polymer on the surface of these cells that we achieved by the genetic delivery of this Apex-2 protein. And if you use EM, scanning EM, zooming in here, if you look, for example, in N, you can actually see this structure forming. It forms sort of a meshwork or a patchwork on the surface of the cells. And again, it arises only in cells that have the gene. I won't go through all the very elegant, I think, chemical characterizations of the method that showed that we were indeed building what we thought we were building in terms of the chemistry, but I'll get right to the function to show that things were functional. What you see here is a pretty interesting assay. The yellow bars and the bright spots in the middle column are gold electrodes, and we had cultured living neurons on top of these gold electrodes and carried out this conductive polymer synthesis. And then we removed the water, dried them out. The cells are now no longer alive. There's nothing conducting anywhere except what they might be able to have within them. And in the upper right, you can see only when we have all the required components together, the presence of the aniline as well as the Apex-2 gene, only then, as shown in the green bar, do we have a current voltage relationship between the gold electrodes. So we're actually creating, indeed, a conductive structure, a polymer structure, in a genetically targeted way. So this was very interesting. We also decided to take this to the mammalian system. This is beautiful work from Yoon Kim in the lab. By the way, Jia Liu spearheaded this work with the broader team, and he's now at Harvard. Yoon Kim in the lab here at Stanford carried out very difficult and challenging experiments where he actually patch clamped individual cells in a living slice of mouse brain while this assembly was happening. And he saw, I won't go through all the details of this, but please check it out if you're interested. We actually looked at a couple different polymers. The middle row is a polyaniline conductive polymer, but in the lower row is a insulating polymer, PDAB. And we saw opposite direction effects on the excitability of the cells that were exactly as predicted from the conducting or insulating polymer properties, changing the spiking of the cells. And we were able to even track this in the same cell before, during, and after the polymerization happened. And then finally, together with Kang Shen's lab, we took this into behaving animals, in this case C. elegans, and we targeted this approach in a number of different ways. First, targeting the pharynx. You can see in the upper right in panel C, over time, the appearance of this polymer deposition is shown with the white arrowheads on this pharyngeal structure in the animal, and we saw a specific change in the pharyngeal contractions that was in the direction expected from the creation of a conductive polymer. And we also saw very specific and appropriate changes in the locomotion pattern when we targeted excitatory or inhibitory neurons that are involved in locomotion. If you look in the lower right, whether we targeted the insulating or the conducting polymer, we saw opposite direction effects on locomotion. So cell type specific creation of a functional structure in a genetically targeted way. So that's what I wanted to share with you. I think maybe you can get a flavor of some of the excitement that we're all feeling, that bringing chemistry to complex intact systems has been really exciting, and we think the sky is really the limit. So many exciting things to do. You can imagine many of them. Just to give you a flavor of where we might be looking in the future. We want to generalize the GTCA approach to diverse substrates or enzymes or reaction conditions, different kinds of triggers, different neural system preparations. And more broadly, we want to link these sorts of approaches, everything I've talked about today in terms of the chemical tools. We want to link these to general, fast, wide field, like in the dissociation experiments, genetically targeted exchange of information, assessment and control within the behaving neural system, linking chemistry to these very high level but single cell grounded interactions with complex systems. I want to thank our very amazing team of students and colleagues that have made all this happen over the years. Our collaborators are shown in the upper right, and the students and postdocs at left. I'm very grateful to them and all our funding agencies. And thank you very much for taking the time. If you have any questions, if you want to explore anything, please just shoot me a note. We're running STARmap workshops. We're working on automated STARmap systems, if you're interested, that take all the microfluidics and the spinning disk and all that and bundle it up into a system. And so let me know if you're interested in that as well. We'd be happy to help. We run free workshops. Anybody can come and take them. So just shoot me a note. And I'll wrap up there. Thank you very much.
Thank you, Dr. Deisseroth. Our next speaker for this webinar is Dr. Mark Brown. Dr. Brown is an engineer and director, systems division at Andor Technology. He continues to be engaged in product management and development and retains a keen interest in biophysics, optics, imaging technology, and applications in the life sciences. Welcome, Dr. Brown.
Thank you, Jackie, and thank you, Karl, for an extremely illuminating and exciting talk. I must say, you've shown me things I had barely even considered possible, so thanks for that. What I want to do is take a few minutes before we advance to questions with a few slides from Andor Technology as sponsor of the meeting. I'll focus on the overlap between the scientific frontiers presented, or some of them in Karl's presentation, and how commercial optoelectronic and software technologies contribute to further and deeper studies in neuroscience and other fields where high-resolution imaging and in situ sequencing can be of use. I guess here we go. So in particular, I'm going to discuss two important confocal imaging technologies and how detector properties and scanning impact their performance. I'd also like to describe briefly the Andor Dragonfly, a high-speed confocal platform, and how it's helping neuroscientists discover, quantify, and deepen our understanding of the biology that underlies all of this. So let me start by reminding you, if you already are aware, and if not, introducing the two forms of confocal technology. So on the left, we have the single point scanning technology, so-called laser scanning confocal. On the right, we have the disc scanning technology. The former relies on sequential scanning of a single laser beam in a rasterized fashion through the specimen. A conjugate detector pinhole delivers optical sectioning, rejecting out-of-focus light and collecting fluorescence from a single voxel at a time. The detector will be a point detector, like a photomultiplier, as shown here, or similar, or possibly an array of these. In contrast, laser spinning disc creates and scans hundreds of laser microbeams or beamlets through the specimen in parallel. The excitation pinholes, shown here on the bottom, are employed in the fluorescence return path to achieve optical sectioning. So we use them to project into the specimen and also to receive the fluorescence. And then a dichroic mirror is used to deflect the beam into a detector. The resulting fluorescence from laser spinning disc is detected through an area detector or a camera. And the scanning methods and detectors define the performance of both instruments to a large extent. Perhaps the key differentiator here is the detector. So, as you probably know, Andor cameras are known for excellent performance and high quality construction. So we introduced the vacuum-sealed deep thermoelectric cooling and introduced water-chilled cooling in the beginning of the 2000s. We pioneered EMCCD technology and led the field with scientific CMOS. Our latest generation detectors are exquisitely sensitive with industry-leading QEs, quantum efficiencies, and low read noise. This makes them excellent detectors for Dragonfly, which is our spinning disc confocal microscope that I'll talk about in a few moments. But I just want to draw your attention to the relative sensitivity of our back illuminated sensors. This is for the electron multiplying CCD on the left, and here for the current generation of scientific CMOS sensors, which are also back illuminated. In contrast, here you have the best performing detectors for photomultiplier tubes, gallium arsenide phosphide and gallium arsenide. So I just want to briefly tell you a little bit about Andor Dragonfly. Dragonfly was born from many years of confocal and spinning disc experience. We've been in this business for almost 20 years. The unique pinholes and microlens patterns were designed to allow you to deal with thicker and larger specimens. The whole area, as Karl has mentioned, has been opened up by the interest in intact biological systems. And so the need to be able to handle larger and thicker specimens was central to the design requirements. We integrated deconvolution with GPU acceleration into the platform to make it a routinely used tool. Those of you who know of Jim Pauly's Handbook of Biological Confocal Microscopy will know that years ago he recommended that this should be the case. We integrated stitching in to handle large volumes, and we include multiple imaging modes in this platform to allow you to achieve resolutions from the nanometers in localization microscopy to millimeters in high-performance confocal imaging. So I'm going to show you a few examples now of applications that have been that benefit from this technology. So in this first case study, is an example from the Blampede lab at UMD in Baltimore, in Baltimore. In this case, they use confocal and super resolution modes in Dragonfly to study NMDA activation in cultured hippocampal neurons. They also used Imaris, an Andor software product, to map synapse position, note that's position relative to the soma, and to study miniature spontaneous calcium transients, MSCATs as they're defined here. And they observed positive but weak correlation between MSCATs and distance from the soma, as illustrated here. Each one of these spots is a synapse, and the calcium transients were studied at these locations. In another example, just switching to explain a little bit about, or at least to illustrate what clarity can achieve in whole cleared brains. Obviously, Karl has described this in some detail, but I just want to say a few words from our perspective. Dense tissue penetration has always been a limiting factor for microscopy. Several techniques have been developed to clarify tissue. Clarity is clearly an important development, as Karl has outlined. And it exploits electrophoretic tissue clearing for speed, otherwise known as active clarity in this case. And clarity retains fluorescent stains, e.g. fusion proteins, allows immunostaining because the hydrogel is nanoporous, and this makes it applicable not only for structural imaging, but for probing tissue for proteins and their precursors, mRNAs. This is so that cleared tissue can be much more deeply probed than obviously the native tissue. And in this case, you can see Thy1 GFP labeled cortical neurons to a depth of 4 or 5 millimeters being shown here, and then rendered in Imaris here, which is excellent for multidimensional visualization. Karl made reference to expansion microscopy, at least in one of the slides. Here is a data set that's been acquired from an XM sample captured with Dragonfly, and again, rendered in Imaris. There is a video behind this, but it's not playing, unfortunately, at the moment. And the results delivered a perfectly stitched sequence in which the neuron hippocampus is clearly visualized. So this is, and the density of these structures is quite amazing in my view. So this is a powerful tool for visualizing large and deep structures. So last point I want to make here refers to the use of STARmap. And we've been working with a number of teams on the use of the STARmap protocols for in situ sequencing. In this case, this is a picture of a microfluidics setup with a flow chamber here on the microscope stage. Here is a Dragonfly on the left-hand side. And what we've done is to, excuse me, having a bit of slide problem there. And what we've done in this case is to I'll restart. Moving on to in situ sequencing, as we've heard from Dr. Deisseroth, STARmap sequences single-cell transcriptomes and mRNAs directly within tissue. So it retains spatial information at a single cell level, which is key to understanding the relationships between cells, their environments, and the circuits they might produce. In this schematic, we illustrate a closed-loop microfluidic system which can be directly automated to operate with Dragonfly and our Fusion acquisition software. Automatic sequencing of the labeling, incubation, imaging, and wash cycles allow the system to function autonomously to deliver multi-round data necessary for in situ sequencing. Here's a typical data set that we've acquired with collaboration from Abbas Rizvi at the Columbia Zuckerman Mind-Brain Behavior Institute. And here you can see 6 rounds of imaging in 4 channels of one single field or one volume. In the next slide, we show the steps from these volumes to achieve cellular identity by localizing mRNAs into single cells. So the volumes of data need to go through several steps in a processing pipeline to convert the images into sequenced mRNAs. So first, the volumes of data are registered so that the spots in each image round are correlated spatially, as shown here. This is the registration step. And then the sequence is then mapped onto longer reads to complete mRNA identification. Pipelines are under development for automation of many of these tasks, not least in the Deisseroth lab, but there are also efforts going in for open source activities from groups like the Chan Zuckerberg Initiative, who've developed a workflow called Starfish, as shown here. In this case, we see labeled mRNAs from an inhibitory neuron resident in the spinal cord. Again, credited to Dr. Rizvi. So, in summary, I just want to thank you for your time to attend the webinar, and just to highlight a few of the points that I've mentioned. Detector quality, quantum efficiency and noise in EMCs and DCMOS are already well optimized, and enhancement continues, but these detectors provide you with exquisite sensitivity for single photon or low photon count imaging. Dragonfly delivers speed, sensitivity, and large field of view with potential for deep imaging. Laser illumination tools that we've developed enable super resolution by localization methodologies. Optical tools to manipulate targeted optogenetic stimulation haven't been described here, but are available from Andor, and you can visit the website if you're interested in finding out more. And finally, Imaris is a leading software tool for image analysis and quantification of these data sets, and we continue to develop this product aggressively. I want to thank you very much for taking the time to listen again, and with that, we'll end and hand over to questions. Thank you.
Many thanks to both of our expert speakers for their presentations. We are now going to move to questions submitted by our online viewers. A quick reminder to those watching us live that you can still submit your question by clicking the Ask a Question tab on the right, typing the question into the message box, and then clicking Submit. The first question that I have is, what is the best use case scenario for using GTCAs when it comes to modern psychiatric diagnoses? Do you see it used broadly across all diagnosis types, or specific to a diagnosis like the dissociative state?
Yeah, great question. I would say broadly that everything that I described, it's better suited as a discovery tool than as a treatment tool. Everything that I described is a way of investigating the brain, and that's what we need in psychiatry and neuroscience broadly, is deeper understanding of how the components of the system give rise to the system function. None of what I've described, I think, is suitable as a treatment in itself. But once you understand the core principles, the causal principles, then any kind of treatment becomes more powerful and more specific once you know and understand what you're doing. And this is our broad goal. This is why everything we do is open source and free. You can get any of our tools, protocols, software, everything come for free training. We want to make sure everybody can can use these approaches to make their own discoveries. And then what happens clinically will be as exciting as anything, but small relative to the broader realm of discovery.
Okay, Dr. Brown, there's a question here for you. Any plan to add volume registration module into the Amaris software, for example, whole mouse brain registration with the Allen Brain Atlas?
Well, that's a very interesting question. It's actually the subject of much internal discussion. At this moment, I'd have to defer to the product manager for Amaris to give full comment. But certainly, atlases are on the roadmap, and indeed for some developmental activities, there has been some work on that front. So I'll pass the question on, and hopefully someone will be able to give you a more informed answer.
Dr. Deisseroth, this audience member is asking about your opinion on light-inducible switch-on PROTACs to enable the degradation of proton targets in a spatiotemporal manner.
So we, as we get better at targeting light to ensembles of cells, individually specified cells, but large numbers of them, you can imagine a number of complex things you could do with that light. Going beyond just turning cells on or even off. Beyond that, you could imagine triggering syntheses of structures in ensembles or assemblies of cells. And you could imagine protein level interventions, changes in the bleaching or fluorescence or color emitted by proteins, changing subcellular localization of proteins with light. And so it's a great question. I think now you can indeed start to think about doing that sort of experiment with ensembles of individually specified cells, like, for example, all the cells that are naturally active during a particular behavior. And this has immense opportunity. You can imagine if you're, as we know in the brain, so many cells that have one job, at least one job we know of, they're intermixed and intermingled with other cells that have completely different jobs or that maybe even oppositional. And so being able to pick out cells and deliver light, deliver function, deliver structure to those ensembles at single cell resolution, for example, using the spatial light modulators, is a very exciting future direction.
Okay, unfortunately, we're approaching the end of the hour. It just remains for me to thank today's speakers, Dr. Karl Deisseroth from Stanford University in Stanford, California, and Dr. Mark Brown from Andor Technology in Belfast, North Ireland. Our apologies if we couldn't get to your questions. Please feel free to reach out to the speakers with more specific questions. Please go to the URL now at the bottom of your slide viewer to learn more about the resources related to today's discussion, and look out for more webinars from Science, available at webinar.sciencemag.org. This webinar will be made available to view again as an on-demand presentation within approximately 48 hours from now. We're interested to know your thoughts of this webinar. Send us an email to the address now up in your slide viewer, webinar@AAAS.org. Again, thank you to our speakers and to Andor Technology for their kind sponsorship of today's educational seminar. Goodbye.